everyone. I'm pleased to introduce Dr. Gregory Holland from the SDSU Department of Chemistry and Biochemistry. Dr. Holland did his doctoral work at the University of Wyoming and has been a faculty member here since 2015. Dr. Holland and his research group applies nuclear magnetic resonance methods to investigate the molecular structure and dynamics of complex biological and technically relevant materials. Of particular interest to Dr. Holland are the many different types of silks produced by spiders. Dr. Holland uses NMR to understand molecular structure of proteins that comprise silks and further to characterize these silks with the end goal of synthetically producing them to manufacture robust materials. So Dr. Holland has recently published manuscripts in the journals of Soft Matter, Biomacromolecules, and Physical Chemistry. Today, Dr. Holland's talk is titled Experimental Biophysics and Computational Tools to Elucidate Spider Silk Formation. Please show a warm welcome for Dr. Gregory Holland.
I should be thanking Jose, who I guess is in Spain right now, enjoying Europe. It's nice to be here to tell you guys more about our work. I actually was looking back, the last time I gave a talk on the CSRC symposium was 2018. So a lot has changed. But the one thing that has grown intently in our research is our use of computational tools. I'm an experimentalist. My background is in experimentation and mostly known for nuclear magnetic resonance, but we use a lot of different biophysical tools. But you know, this audience is quite familiar with like artificial intelligence and machine learning, and just computation and simulation has come so far in such a short amount of time, where 10 years ago I didn't trust anything that I did with molecular dynamics simulation, where now today it's really starting to come to where experiment and simulation are coming together really nicely, and hopefully throughout the talk I'll have integrated in how we are using computation more and more to couple with our experiments to better understand whatever system we're looking at. And my lab is most known for studying spider silk, but we have a number of other projects. And I have my acknowledgements up front. Dr. David Onifree, who actually runs the NMR facility, he's an expert in solution NMR spectroscopy. Dr. Kevin Chalik, who's my postdoc, who's an expert in solid-state NMR spectroscopy. So something that'll become clear is when we're talking about spider silk specifically, we are both interested in not just the solidified fibers, which of course those are amazing materials, and we're very interested in those, but we're also interested in the fluid that the spider synthesizes inside its body and how it converts it from the liquid phase to the solid phase. And NMR is really good at looking at both ends of those spectrum. And I have experts in my lab that look at both ends. And so Dr. Onifree is an expert in solution NMR. My postdoc's an expert in solid state NMR. So David looks a lot at the fluid. Kevin looks a lot at the solidified fibers. Dylan Stengel, who I leave up here, he graduated with a PhD in 2021. He works for Neurocrine here in town. But the reason I leave him in here is he was the one who really got us jump-started in modeling, specifically in coarse grain modeling, and then I'll talk about where we started and where we're at with that. Hannah Johnson, who's a PhD student who's set to graduate, who's spent a lot of time doing electron microscopy, but has really focused on solution NMR recently and looking at a concept called liquid-liquid phase separation, which is something that's important in biology in general, and we think is important as an intermediate step in spider silk formation. And then I always have a myriad of undergrads that span from the biology department. I have engineers, I have chemists, of course, all over campus. I've even had zoologists come through my lab as well, because we handle spiders. And then in terms of collaborators, Nathan Gianescu used to be at UCSD, is now at Northwestern. His research scientist, Chris Foreman, who's done a lot of the modeling work that I'll show you today. And there's a lot of collaborators, and these collaborators are from all over the world. Tom Scheibel, who's in Germany, is one of the experts in making a synthetic spider silk, and so he provides us with some recombinant systems. Chris Lorenz, who's another modeler who I've brought on board, the physicist. Galia Develachina, who's at UCSD, and we're doing some work with dynamic nuclear polarization, magic angle spinning NMR, and I'll talk a little bit about that. Reese Garman, who's in our department, who does ISCAT microscopy. I've actually roped him in as well. And then the funding agencies, most of the money over the years has come from AFOSR and more recently the Army Research Labs. We also get a lot of equipment support. I've gotten a number of Europe awards as well to support things like mechanical testing and NMR probes. And more recently, and I just mentioned this, I'm not going to talk about it today, but for anyone who's interested in sort of surface science interface stuff, we have a new project from the National Science Foundation that's focused on understanding the interface of nanoparticles and biological molecules. And so some other funding agencies, we've had some money from NASA in the past as well. And so this is my spider silk intro slide. And what a lot of people don't realize is spiders don't just make one type of silk. They actually make 6 different types of silk in a glue-like substance. So if you've ever seen a web, that's not just one material. They actually use a number of different fibers to construct that web. And all the materials are very impressive in terms of their mechanical performance, but they vary dramatically. So some are stiff like a glass rod, some are elastic like rubber, some are tougher than Kevlar, and they use all these different materials together to form webs, egg cases. Here's an example here. Wrap their prey. So the money we have from Army is for prey wrapping silk, which is this one. This is showing a tensile testing curve for different types of spider silk. These are dragline silks from different species, and you can see that they have similar curves, but they're not exactly the same. The dragline silk is, if you've ever seen a spider dropping down from the ceiling, that's its safety line. It also makes the framework of the web. The prey wrap is this one here, which you can see is much more extensible and is actually tougher than dragline silk. Spiders only use that to wrap their prey. So if you've ever seen a fly in the corner of a web to be eaten later and it's wrapped in a silk, that's prey wrap silk. It doesn't use it in web construction at all. It also uses it to line the egg cases. So it only uses it for those 2 applications. And that's a very interesting material. And you can tell mechanically it's very different than the dragline silk. It actually has some interesting properties where it changes its conformation when it sees water. Another sort of historical thing that I like to mention, and this is actually from Life magazine, during World War II, the US Army actually had sheds of black widow spiders, and they used to collect their silk on hangers, and that's what they used for gun sights. So this was before we had nylon and polymers where we can make 1 micron perfect cylinders. So the military, and I always remind my program officers of this on this work, the military actually has a long history in that. Like the long-range gun sights in World War II were actually spider silk fibers, and then the black widows, which are the ones that we look at the most. And then, of course, the transitions to biotechnologies. This is a slide that, just about anybody can come up with. You can think of things like, sports, DOD, like, high performance materials, biomedical applications like drug delivery. Of course, suturing materials, silk's been used in suturing for a very long time. And if you actually look inside a spider, and this is some nice art, which is made by a former undergrad of mine, and you actually dissect, and we do this a lot in the lab because we look at both ends of the spectrum. So we don't just look at the silk fibers, but we actually look at the glands where they synthesize the proteins that they spin the silks from. And if you dissect and look inside, there's a series of glands, and the glands that make the different silks all look very different morphologically, right? Just like your organs, they look very different. And then, of course, they use the different glands to spin different silks. The other thing I like to mention to people is people always say, oh, spider silk is sticky. Spider silk is not sticky at all. The spider actually comes around and applies a glue-like substance after it's constructed the web. So the silks themselves, none of them are sticky. They actually make a glue as well. And that's another, interesting biomaterial that you can think about, one that we haven't really studied yet. And this is showing the strength of different dragline silk. So if you take a specific type of silk of the 7 different types, and you actually go to different species, they're not exactly the same, right? So they're all good materials. These are all strong. They're all a gigapascal or better, but they're not exactly the same. And we have some understanding structurally as to why that is. And this is just some comparisons to, other materials. The other thing that people always ask me is it really stronger than steel? It has the same strength as steel, but it's 5 times less dense. So it's significantly stronger than steel by weight. Okay. So some interesting facts. So I'm going to mostly focus on dragline spider silk. So that's the safety line, the framework of the web. And these are looking at two of the primary components that make it up. And what makes spider silk very challenging from an experimental and a computational standpoint is the size of the proteins. So they're huge. They're 350 to 500 kilodaltons, right? So they're multiple thousands of amino acids in length, and they're highly repetitive, right? So you don't have a lot of the specificity that you have. So, I always tell people, most spider silks are made about 60% of just glycine and alanine alone. And when thinking about fibers, and this is sort of old stuff of what we know about the structure of spider silk fibers, something that's been known for a long time, and this is doing solid state NMR work to look at secondary structure, is they have a very high beta sheet content. And that's the same is true for silkworm silk as well. And the long-standing hypothesis is that the strength of silks come from this beta sheet structure. And what I've highlighted in color here, and I know it's very hard to see these polyalanine runs and these terminating GA regions, what I've highlighted in color is what we predict from our solid state NMR experiments form the beta sheet structure. And we know this quantitatively, and this is old work. I'm just giving you, just to give you a sort of an understanding of what we know about silk fibers. But, and if you Google this, you'll find like, oh, it has a high beta sheet content. But I always remind people when dealing with dragline silk, the beta sheet content is still only 30 to tops 50% in the structure of the material. The rest of the material is non-beta sheet. And we've been focusing with solid state NMR a lot on understanding those other structures. So there's things like type 2 beta turns that look a lot like the beta turns that are in elastin, 3-1 helical structures, and of course the naive hypothesis is that these turns and helical structures are sort of responsible for the extensibility properties. And it's the combination of these different structures together that give them their superior toughness. And so when thinking about the starting material, so this is what a major ampullate gland looks like up here in the corner. There's a tail region up here, which is where the proteins are synthesized, and then they store the protein in this gland here. And then there's this ductwork here, which is way longer than it needs to be, that comes to the final endpoint, which is the spinneret, which is the organ that it spins the silk from. This solution here is very concentrated, 30 to 50% by weight protein. And so for the biologists and biochemists, I always just remind them 30 to 50 weight% protein. That is insanely concentrated. If you ever play around with this, it's best described as a hydrogel in terms of its material properties. And so what we want to do, is we can hold both of these. We can hold the fluid in our hand, and we can hold the final fiber in our hand. And we can ask ourselves the question of, what sort of things go on to convert the fluid from here to here, right? And that's the hard part. And there's a lot of action which happens in this ductwork. There's lots of chemistry changes. There's a pH gradient. There's shuttling in and out of different ions. There's liquid-liquid phase separation, which we think is important. There's shearing forces, right? You're funneling this fluid down into this narrow ductwork. And then here's some nice MRI images of what the inner workings look like. These are the major ampullate glands. This is looking down the back of the spider. This is after silking it. You can see the contraction of the glands, and this is sort of reconstruction of what those glands look like. So this is sort of what we know about the spinning process. And when I last gave a talk in here, it was right after we had published some cryo-EM images of these superstructures. The silk proteins are not just in there as monomers, but they're assembled into much larger structures, and we were able to determine this from diffusion NMR, and then we were later able to image it with cryo-electron microscopy. And then there's a number of things that happens as it passes from the gland down into the duct. An important one that I'm going to tell you about today is the introduction of potassium and phosphate, which drives the liquid-liquid phase separation process, which we think is an important intermediate step in route to fiber formation. And then something that's been more well known for a long time is that there's a pH gradient as you go from the gland, where it's closer to pH 7, it gets more acidic as you move down the duct. And then, of course, there's shear forces as well that contribute to the formation of the fiber. The other important thing, and these are the two primary proteins, again, that make up the dragline spider silk, is that you have the repetitive region that forms the core, but then you have a non-repetitive termini region. So this is C and an N terminus on both mass 1 and mass 2. And this is highly conserved across not just dragline silks from different species, but highly conserved amongst the different types of spider silk as well. And there's been some quite a few papers that have come out in high-profile journals like Nature that have shown that these termini regions are actually well-folded helical bundles that are pH sensitive. It's so that there's this clicking together of these different proteins, and that this could help explain how the beta sheets get aligned and how the silk fiber is formed. And this is project objectives taken directly from my Air Force proposal, and I just want to tell you about how I'm going to walk you through this. Like, so whenever dealing with anything in nature, all these natural materials, what nature is really good at that we're not so good at as humans in the lab is being able to control hierarchy, right? Being able to control structural hierarchy. And when I think about this from an experimentalist and also in a computational standpoint, you need to be able to incorporate tools that get you across these different length scales. And so, of course, as a chemist, right, the one, and especially as an NMR spectroscopist, the one that I spend the most time living in is this one down here, the atomic, right? And I'm going to spend a lot of time talking about, you know, individual protein units. This is using molecular dynamics, coarse grain, as well as atomistic simulations. So coarse grain simulations to look at these huge proteins, and atomistic ones to look at some of the finer details. And then how we're coupling our NMR data together with computational tools, Talos N and chemical shift Rosetta. So chemical shift Rosetta is part of the Rosetta simulation package, where the Rosetta simulation is actually restrained by chemical shift information, which we get from our NMR experiments, and then we can get some structural information. I'll spend less time talking about this objective here. I'll talk a little bit about this third objective, and this is this idea of liquid-liquid phase separation. Condensates form of silk proteins, and this is driven by the introduction of the potassium and the phosphate. And this is something that you can mimic in the lab very easily. And NMR is very amenable to looking at these protein dense phases and protein dilute phases, because you could just do this phase separation process and then look at the two different liquids in an NMR tube. And I'll talk to you a little bit about progress in solid state NMR, and this is understanding sort of final fibers, and then some stuff with microfluidic spinning, which we're just beginning to start. I don't have any results yet, but that's something that we're working on this summer. All right, so this gets into the simulation. So when you're dealing with these huge proteins that are, you know, 2,000 to 3,000 amino acids in size, and you want to simulate the entire protein, you can't do this at an atomistic level, right? You can only do this at a coarse grain level. And one of the most common force fields to do this is the Martini force field, and this is one that we've been using a lot. And the Martini force field basically maps to beads. Right, so something like water is actually represented by a bead that represents 4 water molecules, right? So you don't have atomistic details. And when dealing with amino acids, you know, depending on the complexity of the amino acid, it may be represented by 2 beads or 3 beads, or maybe 4 or 5 beads tops if it's something that has a very complex side chain. And so, of course, you lose some of the atomistic detail when you do this, but you gain a 2 to 3 water tube magnitude and speed up compared to atomistic simulations. And so we use these Martini simulations to do the whole protein, and then we do atomistic simulations to do smaller things. For those of you who are interested in simulations and sort of Martini, I would say, from not even from a protein standpoint, but Martini's had the largest impact probably in the lipid world. So it's really, really good at simulating the lipid bilayer for those of you who are interested in those sorts of problems. And so we permit simulations on larger systems, and then you could do things like back map to atomic structures, and we do that as well. And so these are some results on full length protein sequences. And what's known, and I'll get into this a little more about the major ampulate spider silk proteins or the drag-line spider silk proteins, is they look very intrinsically disordered from NMR spectroscopy. And I'll talk to you about what that means. And so when we start with a protein chain, we actually start with a completely random conformation. And we've done this for 8 different random conformations for MASP1, which is the first spider silk protein. And then we've allowed them to go through this Martini simulation. Now, in terms of computational time, you're talking about a month. So what I'll show you today is, is over a year of computational time putting together some of these simulations. And when we first started doing these simulations, and you can see this is the first couple hundred nanoseconds of the simulation, and we're running these out to a microsecond, and you can see we're tracking the radius of gyration during this process. And when we got these end results, and I'm going to show you some small angle X-ray data that's been published in the literature, but we realized that it was collapsing to a radius of gyration that was on the order of what they had seen in silkworm silk protein. So that was promising because it's a very closely related, similar size silk-like protein. But what was emerging in all these simulations is this consistent sort of tubular, worm-like micelle almost type structure. And like I said, we've done these for 8 different ones. This is only showing 3 of them here for math 1. And this is showing what a movie of what the protein looks like, right? So we call these the tubular type tertiary structure. And what's interesting is the certain partitioning of the different amino acids in the structure. And so, the beta sheet forming region is this polyalanine, which finds itself buried in these structures. So the polyalanine is shown in pink, and you can see the pink is mostly buried in the core of the protein. While these large R group polar amino acids find themselves to the surface, which is not really surprising. So what's shown in silver are tyrosine rings, and what's shown in tan and glutamine. So these polar amino acids find their way to the surface. The polyalanine finds itself buried in the core of the protein, and overall it forms this sort of tubular tertiary structure. And we've also done these simulations for the second spider silk protein, which is a bit different than MASP1 in that it has proline in it and a couple other amino acids, but it also has the polyalanine runs, similar to MASP1. And this is showing these sort of tubular-like structures. And the tubular structures have diameters of about 3 to 4 nanometers and contour lengths about 50 nanometers and an end-to-end distance that's 15 nanometers long. And our initial comparison we were making for validation was sort of this silkworm small angle X-ray data, which is really the only thing that will tell you about sort of the globular shape. In terms, you can quantify sort of the amino acids which are on the surface versus those that are buried, and you can see this is surface analysis of the 2 different proteins, MASP1 and MASP2, and you can see tyrosine about 85% is found on the surface. If you look at alanine, only about 36% is on the surface. Most of it's buried. Arginine is mostly on the surface. Some similar stuff here. Of course, in MASP2, you also have proline, and so a lot of the proline finds itself on the surface. And so we have some NMR measurements about dynamics, and this is a nuclear Overhauser measurement. I know you guys don't know what that means at this point, and I'll get into it a little more here. But basically, the stronger this signal is, the less dynamic the amino acids are, and you can see these negative NOEs indicates a high degree of flexibility, and the polyalanine actually has NOEs near 0, which means they're much more rigid. So qualitatively speaking, this was in agreement with this idea that the polyalanine is buried. But the nice thing is that there's been quite a bit of really good small angle X-ray work that's been done. We're actually working on our own as well. And I know these guys, Dick Altari and Volrath, they're a group at Oxford in England. And so, we got some of the data from them, and we combed through their literature and started looking at what the small angle X-ray said, and using that to sort of validate some of our models and how we can sort of, manipulate our model in order to better match the experiment. And this is what SAC's profile looks like. You basically get scattering intensity versus Q space in inverse nanometers. This is for different types of silk. The MA one is the major ampulate, the dragline one. That's the one that we're comparing to in our models. And the other interesting thing is what's shown in here, which is a Kratky plot. That's the inset in here. And you can see this sort of bump feature at the beginning here, and then this dynamic tail. And so this dynamic tail basically tells you that this thing is definitely highly flexible. But this bump feature at the beginning of the cracky pot looks like a lot like this here, which is consistent with something which is partially folded. Not completely unfolded, but not completely folded, but partially folded. And we knew the RG for this was 13.3 nanometers. And we knew from the SAX measurement that these MASP1 proteins are present primarily as dimers and that they were partially folded. And we had some information about the anisotropy. So they were highly anisotropic, and this is a measure of RG over the max Euclidean distance, where the anisotropy is on the order of 0.3. So the nice thing is you can actually calculate small angle X-ray profiles from your models. And we're using a method called FOX-S, which uses the Debye formula to basically calculate this. And so one of the first questions is this dimer idea, and I brought this up at the beginning, that these dimers were formed, and the dimer of the terminite groups has actually been solved by X-ray crystallography, so just a piece of the dimer by itself. And so you could go ahead and bolt together two of these spider silk proteins and start with something that was completely intrinsically disordered, right? So completely unfolded, and that was our starting state. And then you can calculate, you could look at things like the radius of gyration and compare that to what you get. And what we find is that the completely collapsed structure, you know, at 1 microsecond simulation time is way too collapsed compared to what the small angle X-ray tells us. But if you actually average the 100 nanosecond structure, so if you look in the 100 to 150 nanosecond range, so it's not fully collapsed into this tube, but it's not completely unfolded, it's somewhere in between, and you have a dimer of those that are bolted together, you find very good agreement with the small angle X-ray data. So we you know, basically have a model that at least in terms of like overall global parameters seems to make sense and compare with the experiment. The other interesting thing is that selective partitioning of the certain amino acids, the polyalanine variety, the certain polar amino acids on the surface, actually emerges very quickly in the Martini simulation. So, and that's just showing here. You can see this is highlighting the tyrosines on the surface. The selective partitioning is already present in this 100 nanosecond to 150 nanosecond simulation. And so we have dimers from 100 to 150 nanosecond. They match the published SAX data, gives us a correct RG, and gives us a correct RG over the max Euclidean distance of 0.3. And so that, in a sense, makes sense. And so this led us to this study here, which was really quite interesting. And one of the things is that you have this protein sequence, right, that is block copolymer-like looking. You have these polyalanine blocks, and you have these GGX blocks, and these polyalanine blocks, and it looks like a block copolymer. And so one of the nice things about simulations, and this isn't so easy to do in a laboratory experiment, is that you can start, you know, knocking out certain amino acids, moving around different blocks in the sequence and saying, what about the sequence is giving us this? And is there something about the sequences which is giving this tubular geometry? And I know there's a lot on this slide, but I'll just walk you through some of it. And so we looked and we started with some very simple things like polyglycine, polyalanine, and thinking about the shape factor, and right, so this degree of elongation that you have, because this is a very tubular shape, and you get the RG, you know, you could get that from the small angle X-ray calculation of the model, or even your molecular dynamics simulation will give you RG. But then there's ways to get the hydrodynamic radius, and we've been using a method called HOLRAD to get that. And the ratio of the radius of gyration and the hydrodynamic radius will give you an anisotropy factor. And this is something that's well known, I'm sure, for the physicists in the room and the computational people in the room. If you have a value of 0.78, that's a hard sphere. 0.9 is a coil. 1.73 is a rod. The bigger this gets, the more anisotropic the shape is. And so if you look at these polyalanine, polyglycine, poly-GA, which looks a lot like silkworm, these things look very spherical. If you take the MASP1, this is the wild type, you can see we have this rod-like shape, right, which is what I showed you before. And of course, that looks more like a coil, a value about 1. If you go ahead and you knock out all the polyalanine and make them glycines, this thing collapses and it's completely spherical. So if you just knock out all those polyalanines and just make them glycine, it becomes completely spherical, right? If you go ahead and put those polyalanines back in, you get the rod back. So then what we said is, you have these polyalanine blocks in this very defined pattern. Let's move those polyalanine blocks around. So we moved them around, put them all the way on one end, and then started redistributing them out throughout the thing. And what you can see is you get something very spherical, and you move them all to one end. And as you redistribute the polyalanine blocks back out into the pattern of the spider silk protein, you get something which starts to come in this shape like this rod-like shape. So this is giving us some information, and this is something that you could never do in an experiment. I mean, there are ways to do knockouts, but not like this. In experiments, and so this was quite interesting. And nothing, no talk when you talk about computation and artificial intelligence is complete without saying something about AlphaFold. And I'm sure everyone's familiar with this. It's gotten a lot of press over the last few years. Basically, this is Google DeepMind's thing where you could basically feed it just the protein sequence and it tells you what its structure is. And it works entirely on database searching and artificial intelligence and machine learning and all this other stuff. But what I'll tell you from the spider silk protein standpoint, and we've gone ahead and done a lot of this where we feed it in there, it feeds back a really beautiful looking structure. But the structure that it feeds back doesn't look anything like the solution phase structure. It looks a lot like what we predict the solid fiber looks like. High beta sheet. You can see the beta sheets for the polyalanine runs, right? This is getting longer and longer. The match 2 looks a little bit more disordered. This may be more relevant to the gland state. But the important thing is that this whole method is relying on something that has determined and solved structures. So something that behaves like an IVP or something which is partially folded, there's no information for that. So you can use some of that information, but it'll spit out the ordered state. So it doesn't spit out what the sort of intrinsically disordered state looks like. But I just thought I would mention that because somebody always asks. Yeah, of course.
So the shape that it spits out, is it similar to when you had the fully collapsed state in the MD simulations? I know you said if you run it all the way to the end, you get this compact shape. Do those match at that point, or
it's even more compact than that?
Oh, it's even more compact than that. Okay.
Yeah, even more compact. It's something that's complete what you expect for a. It basically looks like a completely ordered beta sheet structure.
Okay.
All right, so the one thing I wanted to mention is some collaboration that we got going on with Reese Garman with doing iSCAT microscopy, because the SAX measurements indicated this was a dimer, right, protein, and we got really good results for these sort of partially collapsed dimers of these proteins that are model matched to small angle X-ray. But it was always a question of whether or not are they really present as dimers? What is the distribution of dimers to monomers? And how can you get a handle on that answer? And this is where Reese came in. And so his method is this iSCAT's interferometric scattering method. And basically, I'm not going to go through the details, and he would be better at it anyway than I would at explaining it. But basically, scattering intensity can be used to tell you molecular weight. And he doesn't really use it much for proteins. He uses it to look at viruses, so very big superstructures. And so what they do is it's a microscopy measurement. They can do some calibration, right? Where you look at some things that form very well-known hexamers and trimers, and here's a smaller BSA, and you basically calibrate your instrument and get some information about molecular weight. And so we've done this for the spider silk protein. And what you can see here, this one on the top is the Black Widow major ampulate dough. And if you look at this molecular weight right here, the dominant component here is 564. And actually, this just sort of worked out this way. If you basically take a heterodimer of mass 1 and mass 2 and add them together, that's pretty close. And I'm not going to tell you that it's just heterodimers. It's probably a mixture of homo and heterodimers. And by the way, there's other proteins in there as well. These are just the dominant species, that's worth keeping in mind. And there's probably some monomers in here as well, and then some smaller stuff. The other interesting thing is that the dimer is thought to be held together through a single cysteine bond and this hydrophobic patch there as well. So you can come in with something like DTT, which is supposed to break those cysteine bonds, and you can see this is very much maintained, which is, again, not surprising because there's a hydrophobic patch in there. But definitely there's some stuff in here that you can see, falls out. So there's some sort of cysteine bonds that are getting broken. But this tells us that, the model of it being mostly a dimer seems to be true. What's important about having this data is that we now have some distribution of the complete molecular weight. So you can come in here and sort of fit a few different peaks. We haven't actually done this yet, but where monomers would come in and some of this smaller stuff would come in, and then you could get a distribution of your molecular weights, and then you could use that distribution of molecular weights to restrain your monomer dimer models, your small angle X-ray scattering data. So you can see where we're going with this. This isn't something that I have the answer to today, but we now have a way to basically map the heterogeneity of that dope onto our models, onto small angle X-ray data, because we, in a sense, know the distribution from this. But the dimer is the dominant thing, and that seems to be correct. But there's more to it, right? It's not that simple. Okay. All right. So this is where I'm going to shift gears to what I really do, which is NMR. And as I mentioned at the beginning, we look at both ends of the spectrum. So solution NMR to look at sort of the fluid that the spider synthesizes inside the gland, and solid state NMR to look at the fiber structure. This is actually some amyloid fibrils. And the other thing, for those of you who are interested in sort of neurodegenerative diseases, I see Stanley in the back there. There's a lot of correlation between spider silk and amyloid fibrils, and this is just an example that I happened to grab to show that it's really solid state NMR is probably the premier technique to look at fibril structure. And so we look at that, use solid state NMR to look at fiber structure, solution NMR to look at structure in solution, and this sorts of information are things that you can map onto things like Ramachandran plots and say things about secondary structure, what forms alpha helices, what forms beta sheets, random coils, turns, etc. And this is something that's been known about in NMR for many years. But what's nice in terms of the developments which have happened, which are mainly computational, is the ability in solution NMR to basically determine structures from chemical shift information, which is really quite powerful, especially for some of these more disordered proteins that we have here. And so what we do, for those of you who are not familiar with NMR, proteins, you want to have C13 and N15 isotopic enrichment. And so in order to do that, we feed our spiders isotopes. So I have lots of undergrads who work in the lab, some of them are in the audience. We feed our spiders isotopes, and then, we collect silk. We can collect the silk fibers to do solid state NMR, or after a couple of weeks of sort of different isotope enrichments. And over the years, we've gotten really good at, which isotopes go to where in the protein and target different types of protein by feeding different isotope diets. And this is how we do it. And the other nice thing is that we can actually take the gland out and snip the tail off and snip the duct off and actually transfer the fluid intact in the gland into an NMR tube to try to keep it. I'm not going to tell you this is native because we've removed it from the organism, but it's as close as you can get. And the other question that comes up is like, yeah, how stable is that? And basically it's stable for a couple of days. You can get a couple of days of good data. Okay. This is what spider silk protein looks like. This is a proton N15 HSQC spectrum. This is the first experiment that any protein solution NMR person would run. What's shown on the right here is GB1, which is a well-folded protein, which has very defined beta sheets and very defined alpha helices. And what you can tell right away is look at the different dots, right? So you see a different dot for every residue in the protein. And this is only 8.6 kilodalton, right? So this is a 56 amino acid protein. So this is really small. You can see all this different resolution you get. And here's spider silk protein, right? So spider silk protein is all stacked up in this middle here. And the reason why is, even though they may be forming these tubular coils, They're very, very dynamic. And this looks like a classic IDP. Anybody who's done intrinsically disordered protein solution NMR will tell you this is what an IDP looks like. You don't have a lot of spread in the proton dimension, and you have poor chemical shift dispersion. And this makes things very challenging, right? Because you can see, like, it's like, if I blow this up, like, maybe there's like 15 different dots here, right? And this is representative of 2,000 or 3,000 amino acids, right? So that's not a lot of information. And so what can you do with that? And this is another thing, and Chris, this is just a message for you here. One of the stories in this whole molecular dynamics sort of computational stuff is that we have a really good understanding of the dynamics of these proteins from NMR relaxation measurements. So T1, T2, heteronuclear NOE. And these things look very unfolded. And what we think is going on is, it's unfolded, it collapsed into this tube, and unfolded and collapsed into this tube. And it's this motion back and forth, right? This motion back and forth, which we know the actual timescales of these motions, and these are picosecond to nanosecond motions. And we can measure these with NMR. And what we haven't done yet, and we've been working with Chris Paolini on this, is the molecular dynamics simulation has those motions in them. And you can calculate those motions from the molecular dynamics simulations, get the correlation functions, and back calculate all the NMR relaxation parameters. And then use that to make sure that the model that you say matches the Sachs compactness, does it match the NMR dynamics? That's a question that I have not answered yet. That this is sitting in revision right now, hopefully soon to be published, and the reviewer wants that tie-in. And we're working with Chris on this. And so there's a set of GitHub, Python codes called Spin Relax that basically can take your molecular dynamics simulations and calculate correlation functions and actually get some of those dynamics out. And this may not work perfectly, and this is going to be very dependent on what force fields you're using and et cetera. And we should be able to tie in the dynamics from the molecular dynamics simulation and hopefully tell the whole story. We at least know the timescales of the yes. Yes.
Are you going to tell us what drives that transition? Is it, for example, is the concentration in the glands different than the concentration outside? Are there other components like agents that would reduce sulfurs, sulfite groups, whatever?
Yeah, I mean, at the beginning, I'm going to talk a little bit about liquid-liquid phase separation, right? So there's different ions, right, that are introduced during the spinning process, right? There's a pH gradient, which we know, and I don't even have it here, and we've published, I mean, if you go into a spider and go from the gland and move down the duct, you will find that the pH gets more acidic. If you go ahead and take your dope and start playing around with the pH of it and make it more acidic, you'll grow fibers. So, I mean, but that's only one mechanism, right? There's lots of things going on. There's also shear, right? There's the fluid flow as well. But we think before all that happens, we know that phosphate potassium comes in, which drives this biomolecular condensate liquid-liquid phase separation process, which has been a hot area I'm sure you're very familiar with in biology, that we think is an intermediate step. So we look at all those, right? Like, in a sense, what I'm telling you about here is really back to square one. Somebody who studied something for a really long time, what I'm telling you is we're still working on the picture of the individual monomer, right? These things are huge. Are they dimers? What is the actual structure of a monomer? Then I could say, all right, let's change pH. Let's introduce these biochemical triggers, et cetera. But we still don't have a good starting structure, right? And that's the challenge with these IDPs. Like, what is the structural ensemble representation, right? All right. But hopefully I'll say a little bit something about liquid phase separation here in a minute. All right, but back to trying to get after the structure of these proteins. And so even though like I said, IDPs are challenging, you can actually do pretty good. And this is looking at the HSQC. And if you go from 2D to 3-dimensional experiments, you can actually start to pull apart and assign different residues. So these are AGs and GGs and QGGs, you know, et cetera, AAGs, and you could do these assignments. And then you could do X-detect solution NMR. So the big problem in resolution in IDPs of proteins is in the proton dimension. So how do you get better resolution? Throw out proton. It's what people in NMR never want to do. Proton's the best nuclei, not for IDPs, it's not. So throw out proton and just have dimensions of carbon and nitrogen. What's the problem with that? The problem with that is that on cryogenic probes, they're typically optimized for proton. But there's special probes, this TXI cryoprobe, and we are lucky that we live in San Diego. They happen to have one on an 800 over at UCSD. And so we can do some of these X-detect improved resolution 800 megahertz measurements. And like I said, you know, I don't know, there's probably only 30 or 40 of these around in the world. And we just happen to be in town here and lucky to have those connections to be able to do that. And by doing these experiments over at UCSD, what we've been able to do is, even with those small little number of dots there, we've been able to expand and do 3 dimensions and X-detect and everything else. And we've been able to assign a 15 residue repeating unit in this protein. And this 15 residue repeating unit, you'd say, all right, well, it's only 15 residues, you know, out of a couple thousand, right? But this 15 residue repeating unit shows up 39 different times, and it shows up throughout the protein. So this is a fairly representative unit. And so what we've been able to do, and I'm not going to show you all the data, but we've been able to get the full chemical shift assignment for nitrogen, NH, H alpha, carbon alpha, C beta, and the CO group for all of these. And there's actually some shorter ones that you can count as well. But, you know, it basically makes up about 20% of the core of the protein, right? So that's fairly representative. It's better than anyone else has done to date. So you can go ahead and take the solution NMR data, use Talos N, which again is one of these machine learning, artificial neural network type things, you know, that computational people are always talking about. And we use these in NMR as well. And you can do Raman-Chandran analysis and output a list of phi psi angles. And then you can go ahead and feed this into chemical shift Rosetta. So Rosetta is a simulation platform developed by David Baker's lab at the University of Washington. And he came together with Ad Bax, who's a solution NMR guy at NIH. And they developed chemical shift Rosetta. This has been out since 2008, and it's been refined quite a bit over the last, 15, 16 years. And what it does is it does simulations that are restrained by NMR chemical shift information. And what's interesting, and this is the 15 residue structures, is that you can see they look very unfolded, right? Which is sort of what we would predict. But what comes up a lot is lots of beta turns. You see the occasional helix, but lots of beta turns. And these beta turns are what's shown in blue. And so if you're thinking about how can you get that compact structure that I showed you in the molecular dynamics simulation, right? But how do you make it be compact, right? But it's still dynamic. Like how do you rationalize all that? Well, beta turns can actually be quite dynamic, and they can be quite compact, right? You're forming turns. And so we've extended this to 120 residue structures, and this is showing some of the lower energy structures here. And if you actually do secondary structure analysis, you can see that like there's almost no alpha helix, almost no beta strand, right? But if you look at beta turns and you look at bends, and of course unstructured, but beta turns and bends make up 50% of the structure. But it has nothing that normally rigidifies a protein. What rigidifies a protein are beta sheets and alpha helices, right? But beta turns are the third most common secondary structure. There's 50% beta turns in there. So that explains how it gets so compact, but can also be highly dynamic. It's also attached to almost, 30, 40, 50% random coil, right? So I don't have the result that's going to tell you the MD simulation is going to predict my relaxation numbers, but that's what we're working on. And the story is, just building. And if you actually do, this is Raman-Chandran analysis, and if you do that, what you find is they're mainly type 1 and type 2 beta turns are the most dominant structures. Beta sheets are non-existent, and alpha helix are pretty much non-existent as well. And, this is also worth mentioning that, people for a long time in the literature have called it an alpha to beta transition. And they called the dope state the alpha state. There's no alpha helix whatsoever. And I can tell you that for absolute certainty. So that's something that's been wrong in the literature for years. We can also do atomistic simulations, and there's a lot of different force fields out there. One of the best for IDPs is Charm 36M. This is a modification on that called Charm 36 IDP. And we've tried a bunch of different ones. My student Julian's in the room here. He's going to try some more. There's lots of force fields out there. They're constantly being developed. And it's not a perfect one yet for IDPs. And this is the one that we found matched the best. And you can see beta turns and bends and unstructured population looks very close to the NMR. There's a little bit more helix than you would like with these. One of the problems with these force fields are that they over-determine alpha helices. It's well known that they don't get the alpha helix right. But anyway, this is just another example of how NMR can help develop force fields too. So, okay. So Stanley, back to your question. So here's another one of one of the mechanisms. One of the many mechanisms that goes into spider silk formation is before this pH gradient, which, like I said, we played around with pH and the dope in the lab, and we've grown fibers from acidified dopes and things like that. But before that occurs, that strong pH gradient, there's an introduction of potassium and phosphate. And this is the work of Hannah Johnson, who's in the audience here. And this is something that I would say is a super red hot area in biology in terms of cellular biology. Stanley will be familiar with this. In the compartmentalizing proteins, and they probably catalyze reactions and all these other things. But it hasn't really been brought into vogue yet in sort of biomaterials formation, that the proteins that form, and this could be collagen or elastin, all different types of biomaterials that form structural proteins, right? And so it turns out that if you come and you take potassium phosphate, you can, and this is also as a function of pH as well, you can basically make droplet formation from spider silk dope. And this is an example on spider silk dope. And I know there's a lot on this slide, and I don't have enough time to go through all of it, but I just want to say that our collaborator, Thomas Scheibel, who's one of the world's experts in making synthetic spider silk, realized that when they added phosphate to their recombinant spider silk protein dopes, they got much better fibers. They never called it liquid-liquid phase separation, but now we know you can just go in the lab and do this and do microscopy, and you can tell that you have droplet formation and you have protein condensates forming. And so this is likely an important mechanism. And what Hannah has been working on is basically looking at this. We've looked at it in the case, and this has been published, in the recombinant system, but we're back looking at it in the native system as well. And, you know, what's going on is sort of this competition between stabilizing the protein and destabilizing the protein. You can see, Hofmeister series, if you look at, your potassium and your phosphate, they're over here. What the gland mostly has in it, in its storage state, is sodium chloride, right? So you're shifting this over here and you're increasing protein stability. And when you do this and add potassium phosphate, you can just drive this liquid-liquid phase separation. And so we've been digging through the biology literature on this. And so some of the driving forces behind this are cation pi and pi-pi interactions. And so silks are multi-glycine and alanine. And what we've been spending a lot of time in the lab in recent years are looking at the scarcer amino acids. There's always 5% tyrosine. There's always 5% tyrosine in silkworm silk, the different silks, all the different silks you look at. Well, what is the tyrosine doing, right? There's arginine in there, and a couple percent level. So there's these other, what I would say, amino acids that have richer chemistry to them than glycine and alanine, right? And so we've been starting to target that, and this is the work of Hannah Johnson, who basically is taking dope, she's dissolving it in urea, and then dialyzing it and exchanging it with phosphate. And then you can drive this liquid-liquid phase separation. And the nice thing about solution NMR is it's really easy to put this in an NMR tube, roll the dilute phase out, concentrate the dense phase, and do NMR experiments. It's probably the best tool to get atomic detail on these processes. And not so surprisingly, because this has been found in other systems, some of the largest effects that we see in terms of chemical shifts, the proteins don't change their structure. They look random coiled, but there are chemical shift perturbations observed, and the strongest ones occur with arginine. There's lots of activity with the side chain groups. There's changes in serine. So the polar amino acids seem to be involved and important in this phase separation process. The other interesting thing, and this is looking at NMR relaxation, which is shown here on the left, is that there's a reduction in dynamics in the glycine-rich region. And these dynamics are not the nanosecond dynamics I was talking about earlier. These are T2 dynamics. So these are microsecond to millisecond dynamics that are changing during this liquid-liquid phase separation process. So the glycine-rich domains are getting more rigid in this gel phase that we phase separate. And we can look at the dilute phase, the gel phase, and then we can compare that to the native phase. The unfortunate thing about the dynamics being in this microsecond and millisecond time scale is that's not 5 minutes. Okay, that's not easily accessible in molecular dynamics simulations. So the nanosecond stuff, the picosecond stuff you can look at, but microsecond to millisecond starts to get difficult. The other interesting thing is in this gel phase, this protein dense phase, compared to the gland. In the gland, the protein interacts with itself and does not interact with water. In this condensate phase, it's heavily hydrated. So that something happens that lets water in, because we can see those water contacts. If you look at the red is the gel phase. You can see the contacts with water. That's these here. If you look at the black, that's the native environment, no water. You go to the gel phase, water. And the same is true if you look at algae, no water, water. So there's something with hydration going on here. And this is a work in progress with understanding this liquid-liquid phase separation. And we've done some simulations. This is really new stuff. Atomistic MD, Charm36M, monomers, as well as hexamers. So these are atomistic simulations with 6 greater than 100 residue chains. So this is, quite a bit in terms of cost, computational cost, but we have some really nice models. I can't tell you the whole story because I don't know the whole story. This is what's going on right now. We have the urea phase, we have the sodium chloride phase, and we have sort of the protein dense with phosphate phase. And we're going through analyzing this, and Hannah's hoping to get some really nice information so she can finish up her PhD this summer. But it's just another step in the process. The last thing that I'll leave you with, because I only have a couple of minutes, is some updates on the structure of the fibers, and this is what I showed you earlier, and we use solid state NMR to do that. And I've alluded to the importance for these chemistry-rich amino acids. So what is tyrosine doing? What is arginine doing? Like, what are some of these doing? And so not just in the DOLE phase, but in solid state NMR of the actual fiber phase, we've been labeling tyrosine and arginine and all these other ones and looking at things like side chain interactions. And we've been able to probe and develop and build on spider silk structural model by being able to incorporate these side chain interactions. And we can see these with solid state NMR, so this proline tyrosine interactions. And then again, building on models, this is work with Chris Lorenz, who also did the DOPE different phase simulations. This is a model where we use both AlphaFold and Charm to build this. This is 10 chains of this, and looking at some of these side chain interactions. So, the models that we're able to build for spider silk structure and what's going on in the DOPE phase by combining computation and NMR together are really powerful. And I think moving forward, we're going to be able to get to where we can have a complete structural model for the DOPE phase and the spider silk phase. And that's the end goal. And then the last thing is dynamic nuclear polarization, which is a method where you add a radical to NMR. We do these over at UCSD and use microwaves to excite electrons, and you transfer polarization from electrons to the proton spin. So, electrons have 1,000 times gyromagnetic ratio of a proton, and you can greatly enhance your signal, and you can see the signal with and without. And this is allowing us to do experiments and look at interactions that normally would be impossible. And this is an example of a cation pi interaction in spider silk fibers that we can access by combining dynamic nuclear polarization with solid state NMR. So, the types of interaction and structural information that we can get out of these NMR experiments is really gone up incredibly, especially since the last time I talked to you guys. And then the last thing I'll leave you with just research plans, because people always say, you're going to make synthetic spider silk? Like, that's always a question I get. Like, you guys always talk about, you learn all this information in solid structure, and then you'll be able to solve this in the lab. and we're working on this, and this is a microfluidics chip. Sam Casenyi's helping us out with this, as well as our collaborators in Germany, and we're building, spinning devices and stuff now in the lab. So there'll be more for that, and I'll just let you guys read the conclusions and take any questions.
Thank you, speaker. Questions for Dr. Holland?
So can you explain a little bit again about the water signal when you said in the LLPS and the NMR? Like, what does that mean, or where is it coming from?
So I'll give you my interpretation, because we don't, in a sense, have the complete story. So these water signals, right, which they show up when you have the phosphate in this protein dense phase, and they're not there in the gland phase, is that the protein in the dope is interacting with itself. There's no contact. So these are NOE experiments. So it means that there's no magnetization exchange between, the backbone amides and water in the dope phase. This hydrogel state, I mean, we know there's water in it, but it's not in close, nanometer or less contact with the protein. The protein is interacting with itself. And hopefully the simulations, we're pulling out contact maps and all sorts of different hydration states and stuff in the ND simulations to go along with that story. And hopefully be able to tell you a little bit more about it. But
what you're saying is that the water molecules are packing more densely when it's in that phase separated state around the protein, or?
The water molecules are interacting more strongly with the protein in the liquid phase separated protein. And that's not a crazy idea, actually.
Okay.
It's known in the LLPS community that these protein dense phases, you think of it as being, protein rich, like it must be water poured. It's not, so there's there's not a lot of water in there, but that water is strongly interacting with the protein.
Yeah, that's the part I was trying to understand. Like, is it forming some sort of a water trap or whatever? But yeah, I can't just imagine that increased interaction as it's not a change in density. It's just local change in density, is it?
It's a look, if you think about in like what I can say from the NMR experiment is that there are waters, less than half a nanometer from the protein that are present in that dense phase, and they're not in that intimate contact in the hydrogel starting stage. And it's not surprising. You don't want the protein doesn't want to aggregate. So somehow in that hydrogel state, that storage state where it doesn't aggregate, it's keeping this hydrophobic polyalanine runs to no end that you can never even synthesize in the lab, and they don't aggregate on you. It keeps them from aggregating with each other somehow. So it has to keep water away, right? But something causes that densification. So somehow I can tell you water's getting in it and intimately interacting. Yes.
So in your image, you have a pi bond stacking on one side, and then you have your alanine chains that are kind of loose net there.
Yeah.
Almost as if there's some kind of hydrogen bonding or something in between there.
Yeah. So I think of it, instead of the cation pi interactions drive the liquid phase separation, the cation pi and pi interactions hold the protein together, and the phosphate allows the water in and breaks those somehow, and the water comes in, and that's what drives the liquid phase separation. That's the working hypothesis for something that's, a work in progress. Yeah.
How electrically conductive are spider cells? How do they compare with like amorphous silicone?
They're not very good. There's actually a couple of papers that have actually appeared in the literature that have talked about them being fairly good conductors. And I'm not an expert in that area, but I just know it's kind of been poo-pooed by people who know more than me.
Is there a layer of lipids around the fiber? So
there is.
Lipids a good insulator. I was just curious if you could isolate a single fiber and use it as a transmission line.
Yeah, so people have actually thought about this, and there is a layer of lipids and glycoprotein a thin layer on the outside. There's like a sheath of it. It's only like 20 nanometers or so, very thin.
Has anybody done signal propagation studies?
There's a little bit, I could point you to it. It's not stuff I'm very interested in, but you would be, yeah.
How does the strength of black widow spider silk compare with like the golden orb weaver spider?
It's stronger.
It's stronger?
Yeah, so, I actually have that on.
I thought the golden orb was like the strongest silk. No, not true.
They just make a lot of it, so people have studied it a lot. Yeah, so this is ultimate tensile strength, 1.4, 1.1,
Oh, okay.
and 1. Which are all good. Those are all strong, right? And there's one that's the Darwin spider, which was found in Madagascar. That's like close to 2 gigapascals. And those spiders, they make 25-meter long webs across rivers. So it's even better than that, but good luck, like you can imagine, it's not easy to get those.
Any other questions for Dr. Holland? Let's thank him one more time.